Good morning. Today I'm going to talk about something a little bit different. I'm going to tell you a little bit of a story about synonymous mutations that we found in an evolution experiment that we then discovered had some pretty significant fitness effects, and our process kind of trying to explore that and think about how fitness effects of synonymous mutations, sort of what drives this and how widespread we might expect these kinds of effects to be. So just to start, I'm going to acknowledge that the work I'm talking about is primarily in collaboration with folks at the University of Ottawa. So this is where I did my PhD, and this stems from experiment that I did during my PhD, and then now ongoing work with, at the end bit I'll talk about is with a PhD student of Reese Cassens, who I'm now sort of informally supervising with some of the work that I'll talk about at the end. Okay, so synonymous mutations, just to remind you, or for those of you who haven't woken up quite yet, these are mutations where the nucleotide change does not alter the encoded amino acids. So because of the redundancy in the genetic code, there's lots of synonymous mutations, lots of potential synonymous mutations. And often what is thought, or what sort of the traditional assumptions are, that these kinds of mutations, these kinds of substitutions have little to no effect on fitness. I think people kind of acknowledge that there is the potential for these kinds of mutations to have fitness effects, but I think that the idea is that often a safe assumption is that they have little effect, and we can sort of ignore fitness effects that there may be with these kinds of changes. And one key thing that comes from these kinds of assumptions is tests where we use this kind of ratio, the ratio rate of rate of non-synonymous mutations compared to the rate of synonymous mutations, to scan across genes and locations, sites, to look for places where that ratio is skewed, so sort of outside of what we might expect, giving us an indication that those sites or those genes may be under selection. However, there's lots of pieces of evidence that we've known about for years, and then now in the last few years, sort of more really direct evidence that this is not always, and maybe not often, a good assumption to make. So one of the key things that comes up is that there is lots of codon bias, and this codon bias varies from species to species. So just the particular codons that are used or tend to be used, they're not, there are biases depending on what organism you're looking at. And so this has been suggested that this may be due to selective effects. I'm just throwing up this figure to illustrate that there's variation in terms of codon use across different species. So this is from a review paper where they talked about these potential fitness effects of codon bias and what drives codon bias. So just to give you an idea of what we're looking at, we have all of the codons here across this axis, and then we have a couple of different species here, Saccharomyces cerevisiae and a bunch of bacteria, E. coli. And then this is sort of just highlighting what, this is actually just for, each of these rows is a different gene within that species genome, just highlighting which are the common codons, and you can see that there's some distinct patterns that characterize particular species. There's also variation from gene to gene within species as well. But there are these really sort of, I guess, clear patterns that suggest that something more complicated is going on. It's not just random. There are also some directed experiments that have looked at specifically generating mutants, so through targeted mutagenesis, looking at the effects of those mutations. And some of those experiments have shown that synonymous mutations can have some pretty significant fitness effects. So one really nice experiment from 2010 in Science showed this. So this is the data from that experiment. They were looking at Salmonella. They went in and did directed mutagenesis in 2 different genes. So the one was rpsT, which is a small ribosomal subunit protein, and then a large ribosomal subunit protein. They generated a bunch of single base pair mutants, some of which were synonymous and some of which were non-synonymous. And so this panel here is the synonymous, this is the non-synonymous, and we've got relative growth rate in LB, a rich media. You can think about this as representative of a measure of fitness, and then number of mutants on the y-axis. So just a histogram of where those mutants fall in terms of their effects on growth rate. And you can see that there's a pretty wide distribution in both the synonymous and non-synonymous mutations, clustered kind of around one. This is relative growth rate, so the wild type has a growth rate of 1, and then, so all of these mutations here in this particular example are deleterious mutations. They all decrease growth rate compared to the wild type, but you can see there's pretty wide range of effects here. And in fact, these distributions, the synonymous and the non-synonymous distributions here are not distinguishable from each other. So in this case, at least, it looks like you really cannot make the assumption that synonymous mutations have no fitness effect, and kind of just focus on looking at the non-synonymous mutations if you're trying to understand selection. They looked in this paper also to try and identify some mechanism for what's driving these kinds of fitness effects. And they did find weak correlation between these fitness effects and change in predicted mRNA structure. So they talk about how changes in mRNA structure could potentially alter rates of translation, and so ultimately, you might expect these kinds of mutations to alter expression, and so have impacts on fitness in that way. But again, I'll point out that this was a rather weak correlation, so it didn't really explain much of the variation. I think it was like 15% of the variation or something like that. Right, so then now what I'm going to talk about is work that came out of my PhD that showed some direct experimental evidence that synonymous mutations with large fitness effects can contribute to adaptive evolution. So we had evidence that there was this potential for fitness effects, but we hadn't actually observed this as being a part of adaptive evolution. Okay, so let me tell you about the experiment. So this is actually just one of the populations from the experiment that I talked about 2 days ago. So that experiment where I looked at replicate populations in a couple of different sugars and then complex combination of those sugars. So it just so happened that we found some interesting things going on in one of those populations that happened to have been growing in glucose-limited environment. So just a sort of standard 24-hour serial transfer experiment with Pseudomonas fluorescens. So to give you an idea of what the adaptation dynamics looked like, so this is a plot of that population over time on the x-axis, and then we have a measure of relative fitness on the y-axis. And so it adapted, it increased in fitness, the population increased in fitness over time, and maybe kind of leveled off to some kind of plateau, perhaps some kind of optimum in that particular environment. And then at the end of that experiment, we had sequenced those final populations at generation 1000. And in this particular population that had evolved in glucose, we ended up identifying 3 different mutations in that population. And this is kind of a typical number of mutations. It's not for this particular species in this kind of environment. We typically see somewhere between 2 and 6 mutations coming up over the course of this adaptation. So the number of mutations was pretty typical. But what we saw that was a little bit surprising at the start was that there's this, where is it here? There's this big deletion of 31 genes, and there are mostly genes that are related to flagella development, which is not surprising in these kinds of evolution experiments where we have populations growing in a well-mixed environment. This happens very often, that we see just rapidly the loss of mobility in these organisms. They don't need to move around anymore. They're just being well mixed, and it's not necessary, and it's costly for them to be able to move around. So that's not surprising. We also saw, however, 2 different synonymous mutations, both actually in the same gene. So this is gtsB is the gene that we saw these mutations in. It's a subunit of a transporter, and well, I'll tell you a little bit more about it in a second. But we saw these 2 mutations, and we thought, well, that's kind of weird. We see 2 different synonymous mutations coming up in one gene, and also, so in this kind of experiment, again, we're dealing with really large population sizes, and so we don't expect to see mutations coming up and going to high frequency unless they really have fitness effects. It's just not, the large population size just doesn't allow for things to kind of drift up to high frequency. So we went back, as I said, we just had yes? Yeah, well, good question. I'm not sure whether that's a significant effect or not. It might just be measurement error yeah, that's a good question. I'm not sure if it's a real effect. And I don't have a good reason. Sorry? Yeah, I'm not sure if that's real, and I'm not sure what would have caused it if it is actually a real effect. Okay, so this was just data, the genome sequencing data was from the final endpoint population. So we went back and we did targeted sequencing to try and figure out what had actually gone on in terms of when those mutations that we were detecting had arisen and what their dynamics were in terms of increasing in frequency through the population. And this is what we pieced together. So generations on the x-axis, and then frequency of the different genotypes on the y-axis. So you can see the large deletion comes up first through the evolution of this population. It increases so that it takes over the entire population. And then we have one of those synonymous mutations arising on that deletion background and starting to increase. And then we see that second synonymous mutation actually arising on a different, on that deletion background, so a different clone here, and then starting to increase. And actually, it looks like this. Well, we've got clonal interference going on, and it looks like that second synonymous mutation is eventually going to win out, but we hadn't quite got there by the end of 1,000 generations. So looking at those dynamics, I was then, again, convinced that these are clearly synonymous mutations with fitness effects, just looking at the dynamics here. But we wanted to make sure that really was what was going on, so we did some tests. These are data showing relative fitness of a couple of different strains here. So the solid blue, solid colored measurements here are from the combination of that deletion plus one of the synonymous mutations. So the blue is one of those synonymous mutations, the yellow is the other, actually corresponding to these same blue and yellow on this plot. And then the hashed bars here are showing where we went and actually put those synonymous mutations back into the ancestral background to look at what the fitness effects were in the absence of the deletion. And so you can see that there's pretty clear significant fitness effects just with that synonymous mutation. So this is one of the synonymous mutations, and this is the other synonymous mutation. So they are having Clear fitness effects, yes. How's it measured? So these are done through competitive fitness assays where we just compete whatever the strain, the focal strain against the ancestor strain, and we look at the initial ratio and the end ratio and see how that's changed, and that gives us a measure of fitness relative to the ancestor. Well, the deletion is... So this is the deletion plus the synonymous mutation. This is the deletion plus the other synonymous mutation. Yeah. I think that's sort of within the margin of error. Yeah, I don't know. Those are one standard error. Yeah. Yeah, for all of these, all of the error bars here are one standard error of the mean. Yeah. Yeah, so I think it's within the same range. Yeah. Okay, so a little bit on what the gene is and sort of how it falls into the genome. Okay, so let's focus down here so what we know from looking at this gene in comparison to other genes in other species, well, E. coli basically, that's been explored and understood a little bit more than in Pseudomonas fluorescens, is that what it is part of is likely a glucose transporter system. So the gtsB is the gene where we saw those mutations of interest coming up. That's codes for this part of the transporter subunit. So it's just a piece of this glucose transporter. And it falls within this operon where we have all of those different pieces of the glucose transport system kind of all together. And what we've got up here, so this is just the sequence just showing you where those two, so the two synonymous mutations are both pretty close to the start of the gene. So this is one of the synonymous mutations, is what we've said called A15A, and G38G is the other synonymous mutation. Interestingly, so I've also highlighted another mutation here. This is actually from a different population, another replicate population that was also evolving in a glucose-limited environment. And we found this other non-synonymous mutation in the same gene. So in some of our comparisons, we also looked to see what some of the effects were for that non-synonymous mutation that had arisen in a completely independently evolving population as well. So then they all kind of fall in the same region of the same gene. Okay, so what's going on? I'm going to show you a couple of slides just to kind of characterize what some of the effects of these mutations are. What I've got here on the x-axis are a couple of different strains. So now we have 4 different strains, each tested in now 3 different environments. So glucose environment, which is the environment where they were adapting throughout the evolution experiment, and then 2 other sugars just to kind of, we were interested to see how specialized some of the fitness effects were that we were looking at. Okay, so here on the y-axis we have relative fitness. And so the first 3 bars here are for the mutations in that gtsB gene. The first 2 are the synonymous mutations that we showed before. And then the 3rd bar here is now also comparing to that non-synonymous mutation that arose in that independent population, just to show that actually the kinds of fitness effects that we're seeing here for these different mutations are basically are comparable, whether they're synonymous or non-synonymous. This 4th bar here is a strain where we deleted the gtsB gene completely. So we wanted to just sort of see what impact that had, and it decreases fitness substantially when they're growing in glucose. And so it sort of just shows us that there is sort of this important role that this transporter has when these guys are growing in glucose. And then there's basically nothing interesting at all going on when you're not in glucose. So this is a really glucose-specific adaptation. Okay, so we wanted to see, well, what's going on? Why do we see these key fitness effects? And our first thought was to think about expression differences. So we tested for differences in expression by inserting a construct. So we have basically just reporter genes that are producing a gene product that we can measure in terms of luminescence. And so this is the construct here. So this is the operon as it stands normally, and now we have inserted in the middle, basically, of that gtsB gene of interest, this set of reporter genes. What we see is that, in fact, those mutations did increase the expression of the gtsB gene. So here we have a measure of expression in terms of relative luminescence. So that's a measure of the product, the reporter gene product on the y-axis. And then on the x-axis, we have those different strains again. So this blue synonymous mutation, this yellow, the other synonymous mutation, and the green, that non-synonymous mutation in the same gene. And here's the gtsB, just the regular wild type without any of those mutations. So you can see there's a significant increase in terms of expression in the glucose environment. But again, this is a glucose-specific effect. So there's no significant expression differences going on in other alternate types of environments. So then we wanted to... Oh, yep, that's true. It may actually be a significant difference. I'm not sure we actually tested that. Yeah. No? What do you mean? Mm-hmm. Like, what are you thinking of? Mm-hmm. Mm-hmm. Well, okay, well, so these error bars are just one standard error. I guess if you wanted to actually look at significance, you'd probably want to look at about 2 standard errors. It doesn't overlap 0. What, this one here? Oh, yeah, okay, yep. Yes, no, yes, I agree with that. Yep. In fact, yes, there may be differences in expression here, but you're right. Yep.
But most people would say that's just a leaky promoter.
Yeah.
But do you see the same effect of the mutation?
Yeah. So it's like
So I think, so the key thing here is that there is an increase in expression, and this seems to be a pretty glucose-specific effect in terms of the fitness gains. What's going on here, there may also be significant expression differences, but that doesn't play out in terms of any fitness differences. And so, I don't know, you can interpret it how you wish. So we know that there's fitness differences. We know there's differences in expression, but of course we want to actually directly link that expression increase to the fitness increase, okay? So what we did was we induced increased expression in gtsB just to look for what the fitness effects might be. So to do that, we insert a second copy of the gtsB gene. So in this case, we're just inserting the wild type because we just want to increase expression of gtsB and see if that in and of itself increases fitness, okay? So we put that second copy in with an inducible promoter, we induce expression, and we measure the fitness. So here we have relative fitness on the y-axis. So on the x-axis here we have, this is just the control with no insert. This is with that gtsB inserted with an inducible promoter. The colored in bars here are when that promoter has been induced compared to when it hasn't been induced. And you can see there's nothing going on here. So we haven't been able to actually increase fitness by inducing expression of that gtsB gene. But if we go and do that for the entire operon, we do see increased fitness. So it looks like that increased expression is so it's not clear if we really need the whole operon altogether. We haven't kind of picked it apart and to see whether it's just a couple of genes within the operon that are really important. But it's some set of those genes that all need to be increased in expression before we see fitness effect. And I guess that's not super surprising because these are all subunits of that transporter. And so one would expect that you kind of need all of them or some ratio of each of those different parts to increase together in order to have some increase in actual functional glucose transport. So, and I just wanted to kind of go back to this guy. Whoops. Here, I just remind you that this is the operon here. And it's the gtsB gene where we have those changes. So it's not really clear why those mutations right here in the middle of this operon would somehow, it looks like, be increasing expression across that operon, and then that is translating into increased fitness. But that appears to be what's going on here. We haven't done those experiments. Yeah. So, yeah, I'm not sure. Okay, so we know from this work that I've just talked about, so we see synonymous mutations that arose de novo in an experimentally evolved population that are contributing to the adaptive evolution of that population. And we've shown that it's through an increase in expression that we see that effect on fitness. And another key thing is that this co-expression of the genes that are part of the operon seems to be a key part of that process. There is more work going on to kind of pick that apart a little bit more and try to understand a bit better what's going on in terms of expression across that whole operon. But it's some combination of increased expression across that operon that's important. And so this suggests that synonymous mutations may play a role in adaptive evolution. We still haven't really identified what the mechanism is that's driving that increase in terms of increased expression. So we know there's increased expression, but why is there increased expression? There's been a whole bunch of work that some students in Reese Cassin's lab have been working on to try and work this out, but it's not, we haven't really figured out. There's not one sort of key mechanism that seems to explain what's going on. My hunch is that it may be a combination of different things, so we can't really say that like all of the synonymous mutations are increasing expression through this mechanism, and so it's sort of hard to identify. But let me tell you about the process that we've taken, or that the students in Reese's lab have been taking to try and look at this a little bit more in detail. And I've been involved in a lot of the data analysis for this project. So what drives the variation in fitness? So we just had those 2 synonymous mutations and another non-synonymous mutation that we were looking at from the experiment. What we wanted to do was get a better idea of sort of in general how synonymous mutations might be affecting fitness. So we did something similar to that Lind et al. Experiment that I talked to you about at the very beginning. We created a collection of single nucleotide mutants. These were just a whole bunch of different synonymous and non-synonymous mutations throughout the gtsB gene. And we kind of tried to spread them out throughout the gene and have a range of different synonymous and non-synonymous mutations, measure their fitness, and then look to see if we can find some general patterns that explain the variation in fitness across that collection of mutants. So these are the data from that collection of mutants that we had. Off the top of my head now, I can't remember exactly how many mutants we had, but it was just over 100, I believe. And we had a whole range of fitness effects. So I've overlaid here both the synonymous and the non-synonymous mutations here. The pinkish colors are the bars are the synonymous mutations, and the bluey, purpley bars are the non-synonymous mutations. So you can see here's the range of fitness effects here on the x-axis and count on the y-axis. This is the fitness of the ancestor, the wild type, and you can see we have both beneficial and deleterious effects of these mutations, and they kind of range pretty broadly. If you so this is a bit wishy-washy, but if you kind of ignore these really large effect deleterious mutations down here, so there's a bunch of non-synonymous mutations that basically seem to be kind of loss of function type mutations. If you kind of ignore them, then the 2 distributions are indistinguishable. And then maybe, I guess, more importantly for adaptive evolution, if you just look at the beneficial side here, they are completely indistinguishable. We're not able to detect a difference between the fitness effects of the synonymous and non-synonymous mutations if we're just focusing on the beneficial mutations. And here's just to show you a close-up view of what that distribution looks like. And for people that might be interested in distribution of fitness effects of mutation, we found that this was roughly exponential, as is expected from theoretical models. Okay, so we took those 100-something mutations and their fitness effects, and we tried to see if we could find some variables that would explain variation in those fitness effects. So things that we tested were looking at change in codon adaptation index, distance from the start codon. The reason that this is something that people think about is that there's an idea that in many genes there's sort of this translational, what they call translational ramp, where you kind of want to start out translation slowly and then kind of ramp up your speed as the gene is translated. And so having codons at the start of the gene that are less optimal is kind of adaptively useful, because it allows the ribosomes to kind of slowly start up and ramp up their speed as they translate. So that's the reason we were kind of interested in that. I guess also because we had in the original 3 mutations we had found in the evolution experiments were all kind of near the start of the gene, so that was of particular interest. We also looked at change in mRNA stability, and this was just through prediction. We didn't actually go in and directly measure mRNA stability in these mutants, but we used software to predict mRNA structure and look at the change in free energy, basically, from the wild type to the mutant structure. And so these are all things that people have thought about as being important for fitness effects of synonymous mutations previously. We didn't actually find any significant effects here, really, except for a bit of a weak, barely significant relationship here with mRNA stability. So we saw no effect at all in terms of codon adaptation index change. We saw no effect at all of distance from start codon. We saw a little bit of an effect of mRNA, but it really explained very little of the variation in the data, something like 10% of the variation. Whoops. Right. Okay, so the next step, I guess, these are mechanisms, sorry, let me go back. These are mechanisms that we thought would be important for transcription, part of the transcription process. And we were kind of trying to think about whether there may be some mechanisms. We were trying to piece apart where the important expression differences might be coming up. Is it at the level, at the transcription part of the process? Is it at translation part of the process? And so the next set of experiments that were done, we're trying to identify whether or not it was increase in transcription or increase in translation that were really important in driving expression differences. So what was done was we had again put in a reporter gene, as I had talked about a couple slides back in terms of trying to understand differences in expression, but put that reporter gene in different places in a way to distinguish between increase due to transcription versus translational differences. So if you put that reporter gene here, and after the end of that gtsB gene, the gene of interest, then you're going to be looking for, if you see increases, when you're measuring that reporter gene product, what you're going to be detecting is transcriptional expression differences. If you put that reporter gene in before the end of that gtsB gene, that gene of interest, then what we're going to get is an idea of translational increases. Okay, so we did that. And here we have those same 3 strains that we had identified from the experiments previously. And this kind of basically shows that there isn't much of a difference between the expression increase that we see from the reporter gene in terms of trying to understand transcription versus translation. So this suggests that it is, that basically what we're seeing is an expression increase that comes through at the transcription level, and then that then follows through in also indicating an increase at the translational level. So if it really was just an increase at the translational level, we'd expect to see sort of lower levels of our reporter. This is the reporter measurement of the reporter gene here on this axis. We would expect to see much lower bars for that transcription versus translation. So there's not really a clear difference here to indicate that that's what's going on. But again, and this kind of just reiterates what I said previously, this expression increase that we do see, and here we've tested a bunch of different mutants from that mutant collection in terms of their measurement of the expression of the gene versus measurement of their fitness, and we do see that there's a really nice relationship there. So it really is that increased expression that seems to be driving these fitness differences, but we're still not really clear on what is making that expression increase. Okay, so I already scratched those guys off the list. Because that experiment suggested that it's really likely to be transcription that's driving these expression differences, we were interested in trying to work out whether there were kind of changes in terms of internal promoters. Those synonymous mutations may be changing promoters, making them more or less efficient, and so changing expression because of those kinds of differences. So,
I still don't understand the rationale.
Yeah.
So,
But-
now you measure again the
I
expression
see.
of the same gene where you saw no effect.
Okay.
And
Right.
I understand it's
That's a good point.
between the translation and transcription. I get this.
Yep.
But I don't understand why you're focusing on the gene that you
Instead
showed
of the whole
previously
operon.
that expression has no effect.
Mm,
You need the whole operon to increase fitness.
Yes. Now, and perhaps I'm describing these experiments incorrectly. So now that you bring that up, I have to go back and double check. But I think that what's going on here is when you are increasing, I mean, these mutations are in here. We're not inducing increased expression in these experiments. This is just expression increase as a result of the mutation in that gtsB gene. And so presumably that increase is then generating increased expression through the whole operon in these constructs as well. But I understand your confusion. Yeah. Okay. So we went through and we tried to look for internal promoters and see if there were shifts in terms of the promoters and how that might relate to where the specific mutations had occurred and their fitness effects. So we were able to predict a bunch of promoters in the gtsB gene and actually throughout the operon, the whole operon itself. The one thing that's a bit of a problem here is that we don't have a really good predictor for promoters for Pseudomonas fluorescens specifically. So we were using prediction software that was using an E. coli consensus sequence. So that's not great. So we're probably missing and perhaps not identifying really well the promoter sequences. But this was kind of the best that we could do with what we had. So we did identify a bunch of promoters through this approach, and then we tried to look at the change in the promoter away from the sort of the optimal consensus sequence to see whether or not those changes might correlate with fitness. There seems to be a positive relationship between predicted promoter strength and both transcription expression at the transcription level and fitness measures, but it's not a significant effect. So it looks like there's a trend that suggests that promoters might be important in driving this, but we don't have a really clear significant effect here yet. And again, that may be because we're not looking for the right promoters because we don't have the right information for this specific species, but who knows? So this is kind of our best explanation at this point. Okay, so now I've kind of finished up where we're at with the experimental work in this project. As I said, there's a bunch of stuff going on that's kind of trying to look a little bit more in detail across the operon to see if we can kind of pick apart where expression differences are, excuse me, and so better understand some of the confusion that Christian has noted. That's ongoing. What I'm focused on in collaboration with Angela, who's a PhD student with Reese Casson, is to try and see if we can look for evidence of adaptive synonymous mutations in, I guess, thinking about whether or not is this just a lab-specific effect? Did we just happen upon some kind of weird kind of gene that happens to have a whole bunch of synonymous mutations that could have pretty important implications for adaptation? Or is this something that's widespread? And how can we actually identify that and look for evidence of that sort of thing? So what we wanted to do is kind of scan the genomes that are out there and use some kind of comparative genomics approach to try and look for synonymous mutations that are under selection. Just to give you an idea of why, I mean, we've been missing this and not really looking for it previously, because often the traditional approach when we're looking for sites or genes that are under selection is to use, again, as I mentioned at the beginning of this, this ratio of non-synonymous to synonymous substitution rates. So just right off the bat, we make the assumption that synonymous mutations, synonymous substitutions don't have a fitness effect, and we just ignore them and look at the non-synonymous sites. So in this, what I'm calling traditional approach, you would collect a bunch of sequences for the gene of interest, align those sequences, then you can estimate your rate of non-synonymous mutations, you can estimate your rate of synonymous mutations, and then basically what you're looking for is places where the ratio of those two is significantly different from your neutral expectation, whether it's much higher or much lower, depending on if you're looking at directional selection or purifying selection. So what we have been working on is trying to use a different approach where we don't make that starting assumption that synonymous mutations have no fitness effect. And instead what we're doing is basically just trying to use the idea that if we see the same mutations, the same changes coming up over and over and over in a phylogeny, that is some indication that those are sites that are under selection. And so what we're doing is basically building a phylogeny, estimating the number of instances of repeated evolution of a whole range of whatever mutations that we can find, identify them site by site across a gene of interest, and then look for places where there is more repeated evolution than one might expect. So we build a phylogeny, we reconstruct ancestral states using whatever method you think is the best. So we're trying a whole bunch of different approaches for this to see, how important is it, what approach we use to estimate these ancestral states. But basically, we estimate those states throughout the phylogeny, and then we just count up how many times we see different changes throughout that phylogeny, what are those changes that we see, and then we kind of just tabulate all of those changes and look at whether they're synonymous or non-synonymous changes. So we kind of, for each site, so if this is the site that we're focusing on here, we'll count through the phylogeny and see how many different changes we see from, say, A to C, A to G, et cetera. And so we have a whole bunch of these kinds of, I guess, transition matrices type things for every single site throughout the gene of interest. So gtsB, for example. So as I say, count the number of each type of change at each sequence position and identify changes that happen more than expected. Now, of course, that's the tricky part, because what's our expectation? That's something that we're still thinking about. But at the moment, what we're looking for is kind of extreme events. So if we generate a giant phylogeny and we just count all of those events, and we look, say, at the top 5% of the most repeated changes, what are those changes? So let me just show you some preliminary work from this. This is an ongoing project. But what we've started with is we've put together kind of two different phylogenies that we're thinking about. One of them is focusing on Salmonella typhimurium, and that's because you remember from the beginning of my talk, I showed you that there was this nice data set where they'd generated a bunch of mutants in Salmonella, mutants in the 2 ribosomal protein genes, and looked at fitness effects. So we have a data set that tells us what we expect the fitness effects of a whole range of mutations to be, so we can kind of compare that to what we get from this approach. So we generate this large phylogeny. Basically, this approach will only work if you have lots and lots of data and lots of diversity, because you're basically trying to tabulate a whole bunch of changes. So you need enough diversity and enough strains to really have a lot of those changes to be able to look for extreme events. If you don't have enough repeated changes, then you're not really going to be able to sort of detect differences from one site to another. So I think this is why this approach hasn't really been taken previously, or at least that's my thought, because you really have to have enough data out there, and we're only now having, these giant data sets available many, many genomes of closely related bacterial species that we can kind of pool together and make giant phylogenies and search across genes. Yeah, that does really screw up things. Yeah, so that's something that we're thinking about. At the moment, what we're doing is just focusing on genes where we don't expect there to have been horizontal gene transfer. We're also really, what we've been doing is just throwing things out of the data set if they look weird. That's kind of a terrible, but we're not looking for ones that are weird across the entire gene. We're looking for interesting single nucleotide substitutions. So if the whole gene itself is weird, then that's probably because it's a horizontally transferred gene. But that's something that we need to think about, not just saying, oh, it looks weird and we throw it out. We need to have some sort of actual criteria for thinking about that and identifying that sort of thing.
Yeah, just because in your previous experiments, it was not a single site on the has been changed. You found evidence of multiple sites. So
Okay, so like 2 sites in a gene that have changed, you might expect that still could be through horizontal gene transfer.
I know, but this ELIFE was several substitutions here, no?
In the experiment, we only saw 2 substitutions.
The other ones were just generated.
Yeah, the other ones were generated. Yeah.
But it means they're possible to turn. I don't know whether
Yeah.
they're biotopical, but at least
Yeah. Yeah, we haven't even started to think about pleiotropy here. Yeah.
Yeah, no,
and
pleiotropy side effects, they may not show up in the experiment.
Yep.
So, but you see still positive effects
Yes.
in the fitness.
Yeah. Yeah, so I would say that this kind of approach is, we're going to be missing a whole lot of things, potentially. It's going to give us an idea of things that might be driving adaptation that are really potentially general across different environments, because we're looking at this phylogeny of a whole bunch of strains and species that have been collected from wherever people have collected things from. And so they are obviously adapting to a whole range of different types of conditions, right? We're not trying to control for that in any way. So any of the effects that we might see from this approach are going to be things that are where substitutions are, tend to be important at that site across a range of environments. So it's not going to, it's going to be pretty general in that aspect. Right, so what we do is we generate a phylogeny. We take, often folks that are generating phylogenies with E. coli and other bacterial species centioids, there's sort of a standard set of genes that we use, a set of housekeeping genes. So we just generate a phylogeny that way. And then onto that phylogeny, you don't want to see that yet. Onto that phylogeny, we then put our gene of interest, basically, and then we work out, based on this phylogeny that we are saying is the true phylogeny, what are the changes that have had to occur to give rise to the sequence that we see in the gene of interest. So then we actually go back and we work out what are the ancestral states on that phylogeny, and as I said, go through and count up all of the changes that have occurred. Okay, so we get a data set that this is just kind of a mess of stuff, but let me tell you what we're looking at here. This is not super informative, but I just wanted to give you an idea of what the sort of complete data set is that we now have after we run through this. On the x-axis, we have nucleotide position. So this is data from looking at the Salmonella and related species phylogeny. And specifically, now we're focusing on one of those ribosomal protein genes, rpLA. So this is nucleotide position across rpLA. And at each of those sites, whatever mutations that we see have occurred throughout the phylogeny that we've built, we've tabulated how many times those mutations have occurred independently throughout the phylogeny. And so all of these different colors here, so you can see there's a whole bunch of different mutations that have occurred, and you may have seen multiple back and forth, all kinds of things going on at a single site throughout the phylogeny. So we have On here we have color is indicating the derived state. The shape is indicating the ancestral state. And so we just have a whole bunch of these tabulated mutations. And so we were thinking about, okay, so on the y-axis we have a sort of a measure of a p-value measure. So the higher up we are here, the sort of more unlikely or more unexpected the data are. So things that are lying kind of at the extreme here are cases where we see a particular change occurring across the phylogeny more than we might expect. So we're still kind of trying to think about how best to go through and identify what really counts as extreme and how to kind of focus a little bit on that. But what I will say is that if we take these data that we generated and actually compare it to the experimental data where folks have gone in and done directed mutagenesis, made a collection of mutants, measured their fitness, that it does in fact correlate it does in fact have a significant relationship with the data that are coming out of this comparative approach. Here are two examples that we've looked at least kind of with a preliminary look. So this is the rpLA gene, and then this is the other ribosomal protein that was looked at experimentally in Salmonella, the rpsT. On the x-axis, we have a selection coefficient that was measured experimentally with that mutant collection from the Lind et al. Paper from 2010. On the y-axis, we have a probability of that mutation occurring on that from the phylogenetic comparative analysis that I showed you before. And so you can see that there is a significant relationship here. This is not quite significant here, but you can see that it does look like there's a correlation between the experimental results and this approach. And so we think that's pretty promising. We're kind of excited by that. Something to note here is also that if you run these linear models and you try to look whether there's a difference between the relationship, if it's a synonymous versus a non-synonymous mutation, we're not actually able to see any significant difference. So, well, this is not quite significant, but at least for this particular relationship here, if you just look at the synonymous or you just look at the non-synonymous, you see the same relationship here, same significant relationship. Okay, and then just another example, we're just sort of starting to do this also with the Pseudomonas fluorescens, compare this to the Pseudomonas fluorescens experimental data set too. So we've built a phylogeny of Pseudomonas strains in the same way and then tabulated various, the numbers of substitutions across that for the gtsB gene, that gene of interest. And then again, this is a similar plot from the previous slide. We've got the relative fitness that we've measured experimentally across those different mutants here on the x-axis, and then some probability that comes out of the comparative analysis. And then we have messy but significant relationship here as well. Okay, so yeah.
You might not expect to see that mutant bouncing around in a phylogeny, right?
Yeah.
So in a way, you're kind of forcing your regression through an origin. So then if fitness is bigger than one, if there's anything, you're going to get some sort of relationship. So is the regression, is the slope different from one if you condition a relative fitness as being bigger than one?
I haven't tried that.
You know
Yeah.
what I'm saying?
Yeah.
Like the ones, like what you see, the pattern you see there is if
Mhm.
as the relative fitness gets really small, that's just an unconditionally deleterious mutation,
Mhm.
which you'd be very surprised to see with extreme.
Mhm.
I guess
Yeah.
in
I
fact,
see your
some of those
point,
ones
but
are really interesting when you measure a fitness of a 10% reduction in fitness, and then you actually see that mutation in the tree. It's pretty funky, right? You know, it's almost interesting in and of itself.
yeah. yeah. But again, this is fitness measured in specific lab conditions, and then it's not. Yeah, but if it's only like a little bit bad, then yeah, no. But I'm not sure. I understand what you're saying with the like you're just not going to see those deleterious mutations, or you're not going to expect to ever see them in the phylogeny. But I'm not sure that that actually is a problem. I mean, isn't that the data?
Points like that are lower than 80% drive
Mm-hmm.
your regression.
Yeah.
Like those points to the left of the graph that are basically maybe unconditionally deleterious could
Mm-hmm.
be driving that relationship,
Mm-hmm.
right? Like it's not like
Sure.
exponential fits this data very well.
No, it doesn't. But I guess the general pattern is that if you're seeing beneficial fitness effects or close to neutral, those are the ones you see in the phylogeny. I mean, I think that's still interesting.
I like that idea.
Yeah.
It's almost like there's a mixture of two things going on.
Okay. Yeah.
Set of unconditionally deleterious.
Yeah.
features
Yeah.
that are maybe obscuring your
Yeah. So an exponential model might not be the appropriate way to look at things here.
that either. It just seems to me
Yeah.
all the interesting stuff is the right side of your graph.
Yeah. Yeah. Yeah, no, I understand what you're saying. I think also, like, we've got a lot of these points here that are all along the zero line because we just haven't observed them at all in the phylogeny. And I think that's also because we don't have enough data. Like, if we were to build an even larger phylogeny, maybe we would see those coming up as well.
You could imagine there's amino acid substitutions that would just destroy the protein function, period.
Yeah, yeah.
There's no
Yeah, but what I think is the zeros that we see that we don't see in the phylogeny, they're not necessarily indicating that's not there because it's a deleterious in terms of its fitness. There's a lot of them I think that we're just not observing because we don't have the large enough data set, basically. So we're when they're not there, it's not necessarily because the fitness is low. It's just because we may not have just been able to observe them in the data. So I think that's something to think about. I'm not sure what a good model would be here, but I think that you're right, that it's something to think about a little bit harder. Okay, well, I'm going to finish early. Sorry, folks. Well, maybe this is not a bad thing because we're probably all tired. I just wanted to say what the next steps are in terms of what we're thinking about with this approach, because as I said, we're really just kind of getting started. The comparison with the experimental data set was really just to kind of convince ourselves that it was maybe an approach that might give us something useful in the end. And we're feeling pretty good that there's a pattern that we're seeing that's not just nonsense. It seems to relate to experimental measures of fitness. And so we're going to continue to try and improve and understand how this approach could be applied. So at the moment, Angela, the PhD student working on this project, she's doing a number of things. We're sort of trying to understand how big of a data set do we need to try and really capture these patterns well. So at the moment, we've been trying to put together data sets of, I guess we're at kind of like just below 1,000 different strains and species that we're building phylogenies of. And I think if we're able to expand that, how much are we going to gain in terms of our power to detect patterns? So trying to understand that kind of thing. And then what Angela is going to do in the next little while is try to actually generate a bunch of simulated data and see basically how robust this approach is to a variety of different things that might be going on. In particular, my interest, as you may have seen from my talk 2 days ago, is trying to also understand how heterogeneity and mutation rates can generate some of these patterns of repeated evolution. And so we want to understand how that kind of thing, how much heterogeneity is going to kind of obscure the results that we are hoping to detect in terms of selection across these different sites. So we're going to think about, how does mutational biases and heterogeneity mutation rates impact what we're able to detect in terms of the signature of selection using this approach. And then ultimately what we'd really like to do is take this approach and scan across many genes and try to really get a good idea of how widespread synonymous mutations with fitness effects are. So can we kind of get an idea of how much we have to worry about synonymous mutations as being important for adaptive evolution across different types of genes? I mean, maybe we can look at, different types of genes that we've been thinking about, like focusing on, say, housekeeping genes, or maybe genes that may be involved in more specialized functions, and perhaps there's some expectations that in terms of where we might expect synonymous mutations to be more important versus not. So we're kind of thinking about that and trying to refine our hypotheses there. So if you have any thoughts or suggestions about that sort of thing, I'd be really happy to chat with you about that. But that's what we're looking at doing in the future. We want to get an idea of how important this really is across a broad spectrum of different genes and different species as well. And so I'll leave it at that. That's all I have for now. Thank you.